Record. Hello everyone and hello to Dr. Novarico. Today we from Group 1, Parallel 3, we will present about the derma dermatophytosis in Trichophyton equinum. So my group member is Sonia Edward and also myself, Muhammad Hazim. So we move to the introduction first. Ringworm, which is formally referred to as dermatophytosis, is a type of fungal infection that target the keratinized area of the body, including the skin, hair, and also nail. This ailment happen due to the existence of dermatophyte, which is consists of Trichophyton, Microsporum, and also Epidermophyton species. So one of the example is Trichophyton equinum, which is a zoophilic dermatophyte infecting horses but can spread to other animals and human too. The lesions are mostly alopecia and circularly in shape, and during a varying stage of inflammation and scaliness. Depending on the condition, one of the method best suitable to diagnose this happen to be dermatophytosis. It could be mistaken for infection of the skin that are bacterial, autoimmune, allergic, or otherwise. So it clearly why a laboratory diagnosis is vital for the reaching an accurate conclusion. Various methods available such as microscopic examination, culture of fungus, and molecular like PCR. So next, the objective of this paper is to provide diagnostic protocol for Trichophyton equinum infection by detailing proper sample collection of hair, skin, and crust, correct sampling, transport, and storage, microscopic examination technique using KOH and stain, fungal culture procedure on SDA or DTM media, native culture examination method, riddle slide culture preparation and staining, and comparative analysis of diagnostic result. The standardized protocol will improve diagnostic accuracy and clinical management of animal dermatophytosis. So we move on to the discussion. Firstly is type of sample to be taken. The most suitable sample for diagnosing Trichophyton equinum is include hair pluck, skin scraping, and crust or scales from active lesion. Hair pluck should be taken from the periphery of lesion. Broken hair in this area often contain the highest fungal load. Skin scraping are collected using the sterile scalpel blade to obtain keratinized material from the lesion edge. Crush and scales can also be useful, especially in cases where hair pluck are difficult to obtain. Next. So we move on to the sampling technique, transport, and also storage. Proper sampling technique are essential to ensure accurate laboratory diagnosis. Before collecting, the area should be disinfected with 70% alcohol to minimize the contamination. For skin scraping, a sterile scalpel blade is used to gently scrape the lesion. Hair pluck should be obtained using sterile forceps, ensuring that the root are included for optimal fungal detection. After collected, samples should be placed in the sterile paper envelope or petri dish to prevent moisture build-up, which can encourage the bacterial or fungal overgrowth. The plastic bag should be avoided as they trap humidity and may degrade the quality. For short term, you can keep at the room temperature. If delay in process is expected, you can put in the 4 degrees Celsius of preserved sample for up to 1 week. So next is direct microscopic examination. This is the low-cost method for diagnosing the dermatophytosis. Technique entails taking hairstyle or skin scraping and putting them on the microscope slide, adding the KOH 10 to 20% to digest the keratin, which help to see the fungal element. The KOH solution may be gently heated but not boil. To improve clearing, the slide is then checked for the hyphae and also the arthroconidia using microscope 10 to 40 magnification. Fungus detection can be often enhanced with different staining, such as cotton blue, has known to mark fungal structure and is many times embedded in the lunate phosphor white stain, which is attached to the fungal chitin, increasing visibility when viewed through UV ray. Next. Next we move on to the fungal culture. Fungal culture stand as the definitive method for diagnosing Trichophyton equinum. Fungal diagnosis utilize two main culture, that is SDA, Saboret dextrose agar, together with dermatophyte test medium, DTM. The SDA media required addition of the cycloheximide and chloramphenicol to block bacterial growth and saprophytic fungi. DTM stand out as a diagnostic tool because dermatophyte trigger a color change from yellow to red during testing, which help preliminary identification. During incubation period of 2 to 4 weeks at 25 to 30 degrees Celsius, researchers grow a sample on prepared media plates with white to buff color colonies of Trichophyton equinum maintain a powdery appearance. Looking at the fungi under the microscope help confirm their identity as a specific species. The lactophenol blue stain on the tape mount reveal both microconidium and microcandium. You can see in the picture. So next I will pass to Sonia.
Okay, hi everyone. So I'll be talking about the examination or microscopic examination on the native culture. So what you do is first when you have the fungal culture, like what we always do in the lab, you use your ose and you scrape a little bit of the fungal culture and you put it on a glass slide which you have already cleaned. And then after that you put a 10% KOH, like 1 drop to 2 drops. This KOH basically what it does, it dissolves the keratin and facilitates fungal visualization so you can see better under the microscope. And then you use the cotton blue to basically, it's a dye basically used for mounting medium and dye which stains the fungal element so you can see the morphology better, and it also preserves the morphology. If you don't want to do this way, you can also use another way, which is the tape preparation. Tape preparation is where you take a transparent tape and you place the adhesive type on the fungal culture that you have, and then you put it onto a slide with a cotton blue, and then you can observe under the microscope. So next, I will show you the microscopic image after doing the slide culture. So this is what Trichophyton equinum will look like. As you can see in this picture, it's labeled, so it's easier for you all to identify what is what. So A, we have the septate hyphate, but in this picture, you can't really see the septate of the hyphae. And then B and C is the microcornidia, which is a cluster, which is also known in Trichophyton equinum, which they are usually very clustered together, which you can see in B and C. And D, Trichophyton equinum is one of the species that has Climodoconidia, which most of the fungal species does not have it, which you can see in D. And then E, we have the spiral hyphae, which is also another characteristic of equinum. And then F, you can see the macrocornidia, the large shape of the microcornidia. I've also included a illustrated image so you can further understand what I was talking about earlier. So as you can see here, there's the spiral hyphae, and then the macrocornidia that I was talking about, and then the climidoconidia, and then the septate hyphae. As you can see here, that you can see the septate hyphae. But in the previous picture, you can't really see the septate hyphae under the microscope, but they have the septate hyphae. And then you can see also the cluster of the microcornidia, which is like branches and clustered, as you can see in the microscopic image. Also, it's clustered together. So I hope this illustrated image will make you understand further. And then next, we will be talking about the slide culture method for the riddle culture. So for the riddle culture, what you use is the SDA agar. So you basically cut from your SDA agar, you cut a cube size, which is around 1 cm cube. And then in the cube, after that, you place it into a petri dish. In the petri dish, there will be a V-shaped glass rod with a slide on top the glass rod. So you place the SDA agar, which you already cut onto the slide, the glass slide. And then after that, you take your colony, the fungal colony that you want to experiment. In our case, is the equinum. So you take the fungal colony and you press it on each side of the cube, as you can see in the image. So they take a small colony and they puncture a small part of the colony into the SDA agar. So that's what the riddle culture is. And then you incubate it for a few days. And this is what the culture image will look like after doing the riddle process. So for the Trichophyton equinum, it will look like this. It will be flat, but some may develop gentle folds or radial grooves, white to buff in color. It will have a dewy texture. Cultures usually have a deep yellow submerged fringe and reverse, which later becomes dark and red in the center. So this is one of the cultures that I found online. And then what's the difference between direct and slide culture? Like I spoke about earlier, from the direct, most of the fungal cultures, especially Trichophyton equinum, they possess uh, the same type of morphology. So they most of it might have the spiral hyphae, they might have the septate hyphae. So it might be a bit difficult to differentiate between what type of species they are. But if you do the slide culture, as you can see here, each slide of the different species possesses a different type of culture. So imagine the T rubrum has this white type of culture. Then the metagrophytes has this white culture with a yellow center. And then the violacum has like a purple center. So all of it possess a different type of culture. So it's easier for you to differentiate which and which. So for ours, it's easy for us to identify is Trichophyton equinum because of the color. So ours has like a orange to red and then it becomes dark. So I can show y'all where I got the image from, just in case y'all don't believe me. So as you can see, I got it from this website. It's called the Adelaide, from University of Adelaide. So they did a research about the Trichophyton, as you can see, done by Trichophyton in 1988. So he did about 22 species. And so happened the Trichophyton equinum is one of the species that we did about. As you can see, this is the morphology they found, which is the picture we used. And this is the picture that Hazim used in his slide as well, with his culture as well. So all three of the image that we use from our slide is from this University of Adelaide. So the one that I did comparison as well is also from this. So I took everything and like I made a collage of everything so it'd be easier for everyone to see, and then I labeled it in this one. So yeah, this is basically our topic. I hope y'all understand. Okay, let me conclude our topic. So through the native and slide culture of microscopic examination, Trichophyton equinum can be reliably identified based on its distinctive morphology and characteristics, while direct preparation using KOH and cotton blue may provide general morphology. The riddle culture slide technique offers a clearer and more detailed view, preserving the integrity of the conidial arrangements and hyphal architecture. The presence of smooth shaped macrocornidia and clustered microcornidia along with septate hyphae, confirm the identity of the organism. So I hope you love our topic today and we enjoyed presenting to you all today. Thank you so much for watching. Bye.
Bye bye.